On behalf of my co-authors, I'll be presenting work on where we've studied the clinical significance of CTC enumeration on the Epic Sciences platform in men with advanced prostate cancer. And so historically, CTCs have been shown to be a very strong prognostic factor, both pre and post therapy, and also importantly have been shown to be a valuable response monitoring tool in breast, colorectal, and prostate cancers. And the way that CTCs are typically counted is they're first captured by EpCAM and then shown to be CD45 negative and express cytokeratins. And here we've evaluated CTC number or enumeration on a next generation CTC detection platform developed by Epic Sciences. And the way that this platform works is that blood is first shipped to Epic Sciences. There's no enrichment step. It's deposited onto glass pathology slides. These slides are then stained with the appropriate antibodies. And then every nucleated cell is imaged. And then in silico, those cells that are CK positive, CD45 negative are identified by advanced computer vision algorithms. And I think for any study assessing the utility or value of circulating tumor cells, it's very important to define first the technology as well as the definition used. And so here we're defining a CTC as any CK positive, CD45 negative cell that has a DAPI stained nucleus. Clusters of CTCs are counted as one event, and then we're reporting CTC number from analysis of 3 ml of blood. And we estimate the turnaround time from collection to result to be approximately 4 days. We selected 175 patients that were treated at baseline who are about to start an androgen receptor signaling inhibitor such as abiraterone or enzalutamide. And these patients were treated in either the first, second, or third line settings and all had diagnosed metastatic castration-resistant prostate cancer. Importantly, we found that CTCs were detected in all lines of therapy in greater than 90% of blood samples. Univariate analysis of survival demonstrated that patients with greater than 10 CTCs detected in 3 ml of blood had significantly shorter survival times than patients with fewer than 10 CTCs. And similarly, those patients with greater than 10 CTCs had shorter time on drug than those with less than 10 CTCs. We also analyzed CTC number as a continuous variable and found that CTCs compared with other blood-based measures such as PSA, LDH, and alkaline phosphatase had the highest predictive ability in receiver-operator curves for 1-year survival. And these results also held up in multivariable modeling using Cox proportional hazards model. And collectively, we found that patients with low CTCs had a median survival time of 32 months versus 13 months in the high CTC group, with a hazard ratio of 2.3 when adjusted for age, line of therapy, LDH, ALK, and PSA. And so in summary, we found that using this next generation Epic Sciences platform, we're able to detect CTCs in the majority of patient samples, and that they're a very strong prognostic factor in patients about to start either abiraterone or enzalutamide. Additionally, we found that patients with low CTCs had longer time on drug and suggests that CTC number on the Epic platform could be used to identify patients more or less likely to have benefit. And finally, we believe the results warrant prospective testing of CTC counts on the Epic Sciences platform as a prognostic tool, and then ongoing work will assess the utility of counts as a response monitoring tool.